Okay, so this video we are going to discuss neurons and impulses or action potentials. So let's get started. Well, if we're going to talk about neurons, let's just do a very brief overview on the nervous system. Like any body system, the cells are the most basic unit of life, and cells are arranged into tissues, and tissues are arranged in organs, and organs into the organ system known as the nervous system. But this particular system is really crucial to controlling our thoughts, the way we move, and a lot of the life processes that we depend upon. Not only that, but our nervous system is quick. Now, you can imagine, in this case, we can see that this person's eye is being exposed to bright light. Look how small the black opening, the pupil is. But in a blink of an eye, in a very short amount of time, if you were to dim the lights in the room, the pupil would relax and open up, and this would allow the little light that's in the room to enter the eye. But this happens almost instantly, but a lot of information has to be passed to and from the brain in order for this to happen. And we're going to kind of learn how the cells do this. So let's start talking about neurons. This is the basic unit of the nervous system. These are the nerve cells. You can see from the picture, they have these weird shapes to them. They almost look like trees with branches and roots to them. Well, their function is to transfer electrical signals, or what we call impulses, to and from the brain. We're going to kind of break a neuron down into three main parts. Number one being the cell body, and this is the part of the cell that contains a lot of the organelles you're probably familiar with: the nucleus, mitochondria, Golgi bodies, you name it. Notice coming off of the cell body, there are lots and lots of small branches. We call these dendrites. You'll see what I mean in a moment. By the definition, it says they receive impulses from neighboring cells. You'll see what I mean in a moment by that. Notice how there's a long stretch, a long single straightaway stretch. That's called the axon. And it will carry an impulse. It'll carry this electrical signal away from the cell body. Again, you'll see what I mean by that. You know, I also want to label the tip of the axon, what's called the axon terminal. That's a very important part, and you'll see much later in this video why that's important. But I do want to bring that up just to get you used to seeing it. Well, look at the top of the screen. So here comes an impulse. I'm just using this yellow spiky shaped object to represent an electrical impulse. Well, notice how there's the cell in the middle. Watch what happens. The cell in the middle will create its own impulse. That impulse was created by a dendrite. Like it says, dendrites will receive impulses from their neighbors. So the dendrite just received this impulse from the cell above it. And now the impulse will travel down the length of the axon, and it reaches the axon's terminal. What happens next? Well, the very bottom cell, the dendrites on the very bottom cell, will receive this impulse and travel down its cell. And we're going to learn how neurons do this as we go through this video. Well, let's first talk about the basic types of neurons. There are the sensory neurons, and like the name implies, these are related to our senses. They detect stimuli and they transmit signals to and from the brain. In this woman's hand, there are sensory neurons, related to our sense of touch and everything. And so let's say she's reaching over an open flame, and the heat activates these sensory neurons. Well, a signal will be sent from nerve to nerve to nerve to nerve to nerve, up the arm, up the spine, and into the brain. So these sensory neurons detect our senses. So we have sensory neurons in our eyes to detect light, and that's our sight. We have sensory neurons in our nose to smell various molecules. We have sensory neurons in our ears to detect pressure. We have sensory neurons on our tongue to detect taste. And we have sensory neurons all over our skin to detect the various touches. Well, then there's the interneurons. Let's come back to our woman here. And these interneurons are located within the brain. So here she is, again, touching the heat, and the sensory neurons that we just mentioned in our hand are going to send a signal to the brain. Well, that signal's going to the brain. That signal's going to the interneurons in the brain. The interneurons are going to process this information. So notice that the interneurons are receiving the signals from the sensory neurons. Well, then there are the motor neurons. And what these do is they're going to pass signals to other tissue. So here she is touching the hot flame. A signal goes up her arm into her brain. And then the brain will send a signal back to the muscles in her hand. Well, let's zoom on in. When we zoom on in, here we see a motor neuron connected to various muscles in her hand. And when the signal gets to the muscles, it will cause those muscles to stretch and contract. Now, what does the stretching and contracting end up causing the woman to do? It causes her to jump away from the flame so she doesn't burn herself too badly. So I hope you saw what I mean by that the motor neurons will pass signals to other tissues. In that example, the motor neurons were passing signals to muscles, and they reacted by stretching and contracting. So let's get into how these neurons work. Let's talk about their resting potential. This is when a neuron's at rest. You know, right now, I don't have any food in my mouth. I don't have a mint in my mouth. So the taste buds, the taste cells on my tongue, are at their resting state, their resting potential. They are not sending taste signals to my brain right now. Well, let's zoom on in to this cell membrane of this neuron here for a closer look. Okay, so you can see the top of the cell membrane is the outside of the cell and the bottom is the inside. Well, we can actually insert a little tiny micro electrode into a cell to record its internal charge. And inside of this cell, turns out there's a negative charge because floating around in the cytoplasm are a bunch of proteins, and these proteins tend to have a negative charge to them. Now, is every molecule in the cell negatively charged? No. Turns out the cell is also packed with a lot of positively charged potassium ions, and those are going to be super important in a little bit. Now, despite the positively charged potassium ions, the internal charge is still around negative 70 millivolts. Well, that's because of all the negatively charged proteins. Well, when we do the same thing to the outside of the cell, the exterior, turns out the exterior registers a positive charge because outside of the cell are a bunch of sodium. Na is the symbol for sodium ions, and they have a positive charge. And so what we're going to see is how the sodiums and the potassiums interact as the neuron goes through its action. So let's talk about how the neuron does its action, or what's called an action potential. An action potential, also known as an impulse. You know, the analogy I like to make is if you've ever gone to a sporting event where the whole crowd is doing the wave and you're just kind of watching the wave go around the stadium. It's kind of fun. One person stands and sits, then the next person stands and sits. In a moment, I hope you'll see why I chose this as an analogy to an action potential. Well, I'm going to put this chart up here. You're going to see this chart a lot, but it'd be a good idea to add this to your notes. But you'll see, and we're going to talk about this scale here as we go through the video. So let's get into how an action potential works. Like we said earlier, sodium is on the outside of the cell and potassium's on the inside. Now, the inside of the cell has a negative 70 millivolt charge because of all the negatively charged proteins. Now, for simplicity, I didn't draw the proteins in my picture because there would have just been too much clutter, but pretend that they are there, given the inside of the cell a charge of negative 70 millivolts. And so what an action potential is, the definition is, it's an impulse it's triggered and it moves down the length of an axon. And so let's talk about how the impulse is generated. Well, notice how the internal charge changed to negative 40 millivolts. Now, you'll see what caused that in a moment. Right now, that's really not the concern. But at negative 40 millivolts, what's going to happen is that black doorway, that black sodium channel in the cell membrane is going to open. And once that happens, sodium is going to diffuse from a high concentration to a low concentration, basically from the outside to the inside. And sodium is going to begin to accumulate on the inside. Well, what's that going to do to the charge on the inside? The negative 40 millivolt charge is going to go up. Negative 30, negative 20, negative 10, 0, 10, 20, 30, 40, 50, and around positive 50 millivolts is when that black sodium door is going to close. Well, something else happens. Let's talk about what happens at the back end of this impulse. Around positive 50 millivolts, there's those red potassium channels that just opened. These potassium channels respond to the internal charge. When the internal charge is reached plus 50, they spring open. And I hope you see what's going to happen next. The K pluses, the potassiums, are going to diffuse from a high to a low concentration out of the cell. And they begin to accumulate outside the cell. Well, what's that going to do to the internal charge? The internal charge is currently positive 50. But as the potassium exits, the charge is going to drop back into the negative range again. It actually drops all the way back down to negative 90 millivolts. And we'll talk about this in more detail in a moment. And around negative 90 millivolts, those red potassium doors close. And then this whole process just repeats over and over and over. And I'll hope to show you what I mean by that in a moment. Well, here we have two neurons in close proximity. That yellow spiky object, there's our impulse, our action potential, and it's moving down the neuron. It's down the axon. Let's freeze right there. You know what? Let's go ahead and zoom on in for a closer look. And when we zoom on in for a closer look, we see the cell interior in gray and the exterior is the white area. Well, let me add that there is sodium on the outside of the neuron and potassium on the inside of the neuron. And here's our scale that I referenced earlier. We're going to use this a lot. Now, the resting potential in a neuron is around negative 70 millivolts. Well, let's say we're responding to touch. The physical force at the surface of the cell will actually deform that first black sodium door and cause it to open. So just the pressure from touch will cause that black sodium door to open. Once that happens, I hope you can see what's going to happen. The sodium is going to diffuse into the neuron, and the charge is going to change. With all of these positive sodiums coming in, the charge is going to go up from negative 70, negative 60, negative 50, negative 40, negative 30, negative 20, negative 10, 0, 10, 20, 30, 40, 50, and around positive 50, look on the chart, the scale, the black sodium door is going to close. Not only that, but the red potassium doors open. Well, once they open, I hope you can see what's going to happen next. A lot of the potassium will start to diffuse out. What's that going to do to the charge? The positive 50 charge is going to drop because the cell is losing a bunch of positive potassiums. The charge goes from plus 50 to plus 40, plus 30, plus 20, plus 10, 0, negative 10, negative 20, negative 30, negative 40, and something happens interesting around negative 40. Watch what happens around negative 40. The next black sodium door is going to open and sodium is going to start to diffuse on in. Now, even though sodium is diffusing in, watch the charge. It's still going to drop down to negative 90. Well, I hope you see why that is. There's just more potassium doors open because there's more potassium channels open. Potassium is going to exit in greater amounts and the charge is going to keep dropping. Well, once we get to negative 90, look on the scale, those opened red potassium doors are going to close. Well, now the sodium is the only thing that's entering the cell, and it's beginning to accumulate. What's that going to do to the charge? The charge is going to start to go up because all of the sodium is now accumulating. It's going to go up from negative 90 to negative 80, negative 70, negative 60, up, all the way to plus 50. Look on the scale. What happens at plus 50? The black sodium door closes, and the next set of red potassium doors and channels open. Well, once those potassium channels open, the potassium will start to diffuse outward, and it starts to accumulate outside of the neuron. The charge drops. The charge begins to drop again from plus 50 all the way down, and around negative 40, the next sodium door opens, and even though sodium is diffusing in because there's more potassium diffusing out, the charge continues to fall all the way to negative 90. At negative 90, the red potassium doors close, and the sodium is still entering, and so it's starting to accumulate. That's going to cause the charge to go from negative 90 all the way back up to positive 50. Look on the scale in the upper right-hand corner. At plus 50, that black sodium doorway channel will close. The next set of red potassium channels will open, and we're just repeating ourselves. The potassium begins to exit, and the charge plummets. Well, here's a little simpler animation that, again, I hope you see why I chose people doing the wave at a ball game as a good analogy to an impulse. The person doing the wave stands up and sits down. The next person stands up and sits down. That's kind of like every time sodium comes in, the charge goes positive. And then when potassium exits, it goes back to negative. So the charge jumping up to plus 50, negative 90, plus 50, negative 90, standing, sitting, plus 50, negative 90. I hope you see why I chose the analogy of people doing the wave at a ball game. Well, now that the impulse has passed, I hope you see we've kind of got a problem. The natural state of the cell is now out of balance. Remember, potassium is normally on the inside of the cell, but a lot of it's now accumulated on the outside. And sodium is normally on the outside of a cell, but a lot of it has accumulated on the inside. There has to be something to clean up this mess and reset the cell to normal. The cleaning up the mess, who cleans up the mess? It's called the sodium-potassium pump. In the cell membrane, there's another, there are other objects. I didn't add them until right now. There they are in blue, the sodium-potassium pumps. They've always been there, pretend, but I've been holding them until now to talk about. Well, how do they clean up the mess? They use ATP in active transport to pump molecules from a lower to a higher area of concentration. And so what is going to happen during this cleanup process? Sodium is going to be pumped back out of the cell. The sodium-potassium pump can actually hold 1, 2, 3 sodiums at a time. And with the help of ATP, an enzyme will break ATP and release heat. That heat will cause the sodium-potassium pump to change its configuration to allow the sodium to be pumped out. Not only does it pump sodium out, but it also will pump a couple, 1, 2 potassiums back into the cell. As the heat wears away, the protein returns to its normal shape. The sodium-potassium pump returns to its normal shape, and the potassium is pumped in. Let's repeat that again. 3 sodiums at a time load into the sodium-potassium pump. A molecule of ATP is broken. The heat causes the sodium-potassium pump to change its shape to release the sodium. Then 2 potassiums enter into the sodium-potassium pump, and it returns to its normal shape to bring the potassiums in. So when does this cleanup happen? What happens after the impulse is passed? Where is the sodium-potassium pump located? It's in the cell membrane. But most importantly, why is it important? It restores the initial charge inside and outside of the cell. Let's do that one more time. three sodiums enter into the sodium-potassium pump. ATP is broken, heat is released. It causes the sodium-potassium pump to change its shape, releasing the sodium. 2 potassiums enter in. The sodium-potassium pump returns to its normal shape. The potassium is brought in. You can imagine if all of these are pumping sodium out and potassium in a very short amount of time, boom, they can reset the cell to its normal -70 millivolts resting state. And now the neuron is able to send another impulse should it be required to do so. So that's really the importance of the sodium-potassium pump. Well, here we are again at our 2 neighboring neurons. Again, here's the axon. Here's the axon's terminal, and here's the dendrite of the neighboring neuron. Well, let's continue the impulse down the axon, and eventually the impulse arrives at the axon's terminal. We got to zoom in here and take a closer look. When we zoom in, we come to what's called the synapse. Well, here's the axon terminal on the left. Here's the dendrite on the right. Notice there's a little gap in between the two. That gap is called the synapse. It's the gap between neurons. Well, let's talk about what happens here, the steps that happen here. Well, the impulse will reach the axon's terminal. So here we have a sodium channel opening. Sodium rushes in and the charge jumps up to +50. We just got done talking about that. But now the impulse has reached the end of the axon's terminal. Notice how there are vesicles in the axon's terminal with 3 little black dots in them. We'll talk about those black dots in a moment, but vesicles are little storage organelles. And notice what's going to happen here. This vesicle is about to fuse with the cell membrane. And when it does, it's going to release those black dots. Those black dots are beginning to be released into the synapse. Those black dots are what are called neurotransmitter proteins. Neurotransmitters are released from that vesicle, and they will diffuse across the synapse, and they land on what's called the dendrite receptor, a little landing zone that the dendrites have to catch these neurotransmitter proteins here. And now that the dendrite receptor has caught the neurotransmitters, watch what happens. It will open its own sodium channel, and then sodium rushes in. Like we talked about a moment ago, the charge jumps up to +50. When that happens, the black sodium door closes, the red potassium doors open, and the potassium begins to exit. When that happens, the charge falls all the way down to -90 millivolts. We just got done talking about this. Notice what's happened. The neuron on the right created a brand new impulse. And here we have 5 neurons. Let's pretend that you want to move the muscles in your foot. And so what happens is cell number 1 will make an impulse. The impulse travels down cell 1. Cell 1 releases neurotransmitters. The neurotransmitters are caught by cell 2. Cell 2 makes its own impulse. The impulse travels down cell 2. Cell 2 releases neurotransmitters, which are caught by cell 3. Cell 3 makes its own impulse. The impulse travels down cell 3. Cell 3 releases neurotransmitters, which are caught by cell 4. Cell 4 makes an impulse. The impulse travels down cell 4. Cell 4 releases neurotransmitters, which are caught by cell 5. The impulse is created, and it travels down cell 5. Well, where is that impulse going? Well, we saw this before. Here's a motor neuron sending an impulse to some muscle cells. How do the muscle cells react when they are zapped with that impulse? The muscle cells begin to stretch and contract, and it's what causes movement. So we've tracked in this video how neurons send signals from neuron to neuron to neuron, and eventually how that signal causes your muscles to move and react. And so there you go. There's hopefully a good understanding of how neurons will send impulses and how they communicate with other neurons. If you're in my class, pause the video, try to answer these questions. And thanks for watching.